Much for the invitation and for the nice presentation. It's my fifth time here. So sorry if some things are the same, but yeah. I've been out of science for a while, but now I'm back. So I will tell you a little bit of old story, but this is for the students, so I guess you will appreciate that. So let's start really quick. Okay, so I'm going to talk about spinal cord stimulation for the treatment of the motor symptoms of Parkinson's disease. And we're going to talk also about motor control. This is my scientific interest. So let's start with what is spinal cord stimulation? And I will start saying that spinal cord stimulation is very old. So it's like 50 years old. We have been using it for, if you read like paper from 1967, it's electrical inhibition of pain by stimulation of the dorsal columns. So we have treatment of chronic pain through the electrical stimulation of the dorsal column, which is a part of the spinal cord. So what you do here basically is you put electrodes in the epidural space of your spine and you crank the electricity until the person reports no more pain. This is a pain that is refractory to any regular pharmacological treatment. This is the spinal cord, for those of you not familiar with it. So spinal cord is not just the interface between the body and the brain, but it's also an important computing or integrating structure. So it does computation for us actually a lot, and it's quite tiny if you compare it to the surrounding structures. Like this is like the vertebra, right? And here's the spinal cord. And in more detail, right? We have this gray matter, which is the processing center of it, right? And in blue we have ascending axons, right? That they convey information to the brain. And in the red, we have the opposite, right? So axons that actually convey information from the brain towards the spine and towards the periphery. So it's like a two-way, right, highway for information and for processing, as I said. So this one here, this is dorsal, this is ventral. So this is the actual dorsal columns, right? And they're quite organized. They have a somatotopic organization. So according to the part of the body, right, you can find here the legs, right, the arms, and then more cervical, right. And you put the electrodes here, right here, above it. So, but here's the dura. So you don't go inside the nervous system. You are outside the nervous system, and here you put your electrodes. And the electrodes, they look like this. So these are the ancient electrodes. They are just like a cylinder, and these are the contacts. So here you will have currents. You can have monopolar, just one contact working, or you can have bipolar, two contacts, one positive, one negative, so you have a more localized stimulation. And these are more recent electrodes. They're better because they have more contacts and they are closer to the spine. They're flat, not round like these ones. And you have many contacts, so this is how they look when they are implanted in the epidural space, right? This is the electrodes, and this is the wire that goes to the pulse generator that will generate the pulses, the electrical pulses for the stimulation. And this one here, so it's the same concept. So you can put it in different levels of the spinal cord. Okay, so what do we activate when we do the spinal cord stimulation? So this is a model, a computational model. This is the spinal cord. This is the epidural space right here. And this is the electrode. So you can see these lines. Those are the electrical fields, right? This is pretty much the electricity we deliver when we stimulate. And we know, because of the experiments and because of this modeling, we know that, oh, sorry, this is okay, here's the English part. So we know that we will activate the A beta fibers, the superficial one, the one that are in the surface, and large diameter, preferentially, If I crank more electricity, I will stimulate other elements as well. And the same fibers, but this time in the dorsal roots, right? So the dorsal roots is the entry of the sensory system towards the spinal cord. Okay, so we know what we activate, and now let's see what type of information does these structures convey. And here are the dorsal columns. I just showed you them, right? So what they convey the somatosensory and the proprioceptive information from mechanoreceptors in the periphery. So that's what they do. So they will just enter the spinal cord and they will ascend right through the spinal cord, and this is where we put the stimulation. So when I activate here an axon, right, I will be activating this pathway, and in the medulla it will actually go to the other side and will climb even more to the thalamus and then to the cortex. And specifically it's the primary somatosensory cortex. So, question for the students. So when I do stimulation, electrical stimulation here, what should I feel? What's the sensation? What do you think? Pain? No. This is good for relieving pain, actually. What do you expect if I'm telling you that this goes right to the primary somatosensory cortex? Okay, so here's the answer. Okay, so people feel paresthesia. And paresthesia is just numbing and tingling of, well, any part of your skin. That's what you feel when you stimulate the spinal cord, because you are activating the somatosensory system, right? So this is a very nice plot, because it has information for like 2,000 patients, and they're implanted with electrodes in different parts of the spinal cord. So this is the neck, right here, the thoracic, and this is lower thorax, right? And here you can see which part of the body they report to feel the paresthesia when I stimulate. So, for example, if I put electrode in the cervical part, you're likely to feel paresthesia in the hand, right, or in the arm, right? If I put it in the low thoracic part, right, likely to feel paresthesia in the foot and in the legs, right? It makes sense. And then there is a magical spot right here, this one, T1, T3. But you're likely to feel everywhere, right? So probably you're recruiting fibers that come from below, right? So you are likely to feel paresthesia pretty much in every part of the body. So now that I explained it, spinal cord stimulation, let's go to the second part, which is Parkinson's disease. So, and I'm going to talk about some of the mechanisms underlying the motor symptoms of Parkinson and how we treat them. So it's important to keep in mind that Parkinson's is a neurodegenerative disease. So it's not only affecting the motor system, right? It's not a motor disease. It's neurological. So it will affect, it has many symptoms other than the motor. The motor are very visible, they're apparent, right? And people tend to try those, to treat those. But it's not only a motor disease. So try to put this video here. And this is a patient with classical Parkinson motor symptoms. So tremor, the first, and then inability to start moving. And I will explain why that happens. So when he starts moving, it's very short steps. The woman, what she's doing, she's giving him like a sensory cue for him to start moving. That's something that works pretty good with Parkinson's disease. And this is fascination. So his steps are so short. Yeah, his gravity center goes forward faster than his feet, so he falls down. And this is very important. This is freezing. And freezing is when he started already walking and he wants to turn, he will just froze the gait. And this is the same patient on a bike. Yeah, yes, it is. But there are more to this video. If you can see the lady here moving, there's a lady moving a car. So he does pretty well, right, on the bike. So there's the lady moving the car. Yeah, so you see him very, like, perfect, right? So there are many explanations for that. There is no consensus about why this happened. He was a biker, so he probably has some functional reserve somewhere in the nervous system to do this. But look, when he goes back, he froze immediately, right? So, and here it's important to see that the bike, well, has no friction, right? You're on wheels. And you saw that when he started on the bike, there was the nurse that gave him a push. And apparently that's the difficult part to overcome in Parkinson's, like the start of the movement. So once you're moving in wheels, okay, that's fine. And also, the sensory input he's getting back is different. In the bike, right, most of his body weight is on the seat. But then when he's on his feet, it's on his feet. And the spinal cord will sense that, right? And that sort of freezes him. So we don't know why. I just showing this because it's a very, very interesting video, and it shows also what are the Parkinson's disease motor symptoms. So let's go a little bit to explain these symptoms. And I like very much this review. If you have chance to read it, do it. I mean, you have an interest in motor control. So this summarizes what we understand on motor control, right? So, I mean, most people, or we tend to think that we have like a cell, a neuron in the brain, in the cortex, and that control the muscle, right? And we move. And that's true, but it's a bit more complex than that. So, what I should start saying that when we move, we control many muscles, hundreds of muscles, right, in very precise coordination, right? And you don't think about it. So when you walk, you're not thinking right, about keeping your posture and moving your leg forward in the other way. If you do that, it's sort of, right? I mean, you can text on your cell phone and walk, right? And this is a good example because texting on the cell phone is a different control than walking. Or it's the same, but it's just on different ends. So we have these motor programs here. These are motor programs. And motor programs are like a sequence of movements that have some functional meaning. They serve something. So the most simple is reflex, right? So swallowing your food, right? Another motor program. So breathe, right? Respiration. Locomotion, posture, chewing, expression of emotion is a motor program, or it's thought to be. And eyes movement, and reaching with your hands in the case of primates, right? So we go from the most simple to the most complicated. And the thing with those is that, especially the ones here, they reside or they are generated in the spinal cord or in the brainstem by itself. So both in spinal cord and in the brainstem, we have circuits, hardwired, right? They can just generate this movement just by stimulating the circuit, right? And we know that a while ago already. So you stimulate here, yeah, you can generate one of these, probably locomotion, strong enough, right? You will affect like the motor part of the spinal cord, and so on. So this solves an important problem, right? So I don't think about coordinating my legs when I walk, right? Because the spinal cord does it for me. So the next things I want to do, then if I want to control locomotion, I want to just turn off and on these circuits, right? And this is done by a certain population of neurons here in the brainstem, but the one that select which motor program I want to do, these are the basal ganglia. So this is a very important structure in the motor control. It is so important, right, that our ancestors, like 500 million years ago, they didn't have a cortex, right? This is a lamprey, it still exists. But the first vertebrates, they didn't have a cortex. They have basal ganglia, and they have like local spinal and brainstem circuits, and they could do everything they needed just with that, right? How does this work, right? The basal ganglia select what has to be done in the precise moment. This is very important. This is no joke. Yeah? It sounds obvious, but it's super important, right? So if you're in front of your food, you want to move to your food, right, and eat. If you're in front of your predator, you want to get away from your predator, right? And you have to do the right choice for that, right? You don't want to make a mistake. Right? You don't want to approach your predator. Yeah, you're dead. And the animals that didn't have, the right way to do selection, they died. So, okay, so basal ganglia will select among all of these motor programs which one to initiate. The initiation of the motor program, this is very important. And stop it also, it's important to stop, right? So now the problem is much simpler, but we need something to get the basal ganglia to decide what to select, right? And the basal ganglia in the primitive form, right? It will decide it by sensory input, right? Could be external, right? Food or predator, or also internal. Yeah, I'm hungry, right? So I will look for food. So sensory input here, both internal and external, will feed the basal ganglia and will select an action. So we solved most of the typical functions, or it's called motor repertoire, right? So what we do every day. But then in primates, we have, or in general in animals with the cortex, right? So we have another invention, right? And it's called a cortex, cerebral cortex, right? And the cerebral cortex will also feed the basal ganglia with information, with sensory information, but with volitional information. So I want to move, or I have to study for my exam, right? Or I want to enter a PhD or a master's, right? And that turns into actions, right? So now we have this cerebral cortex that will feed also basal ganglia, but also the cerebral cortex will perform or be crucial in what we call fine motor skills. And now we go to the single neuron here, or a few neurons that actually control directly some of the low or the inferior motor neurons here. So you have actual control of single muscles. So if you want to write, for example, or if you want to talk, right, you will use a lot of cortex doing so. So this will be speech and hand, finger, fine movements. So that's why you can walk, right, without not paying much attention, but you can walk and you can text on your cell phone, and when you text on your cell phone, this is fine motor skills, right? And as long as the environment is flat and there's no obstacles, you're okay, right? If not, then something can happen, an accident. So, okay, so this is the brain, or the nervous system controls the movement. Please see that this is the complete nervous system is engaged in motor control, right? And now I talk only about motor control, like what goes from the brain towards the muscles, right? But there's a component that I ignored here, but it's very important, is the sensory component, right? I cannot move without knowing what's going on, right? And that's the other like side of the coin. And also, well, cerebellum is here, very important also, because it's like a manager. It will supervise that every command that is given, right, is executed properly or not, and will do the right correction for that. So now let's go in detail to the basal ganglia. And this is okay. This is the organization and function of the basal ganglia. This is another review also for Greenland. It's old, but I like it because it's very simple to explain. So this will be the basal ganglia, right? These three structures here. This one is the stratum, and we know the stratum is the most important structure in the brain because it has glycine receptors. Just learned that yesterday. But so this is the stratum, and the stratum is the input to the basal ganglia, right? And here's reverse pallidum, so just keep pallidum, this is okay. Pallidum is the output. So we have an input, we give information, and we have an output, we receive information. And what is the input to the stratum? Is cortex and thalamus. So again, cortex is like the volition, it's also sensory, high order sensory, but it's also thalamus, and thalamus is sensory, right? So again, the same scheme, right? So we provide like our intentions, we provide the sensory information to the stratum, and something will happen here. What will happen? We will activate either of those pathways. So we have a direct pathway, which means a stratum, the MSN neuron, projecting directly to the pallidum, and those are inhibitory. They are GABAergic, right? And we have these other indirect pathways because this MSN project to the external pallidus and in turns to the subthalamic nucleus. Subthalamic nucleus is important because it's target for deep brain stimulation. And in turn, subthalamic nucleus now project again to the pallidum. So, but we start to explain this, we start from bottom up. So the output of the stratum is actually also inhibitory, right? And you see here high rest activity. So the pallidum is discharging, firing all the time, right? When we are not moving, it's discharging pretty much all the time. And it's inhibitory. So it's inhibiting the motor program. So our baseline is every motor program is shut down. And the way I control it is that I release the inhibition over a single motor program or more than one, right? So I can initiate movement. And then when I don't want to do this movement anymore, I shut it down again. And this is all the time. So we break all the time, right? So when I want to start a movement, what I do, I inhibit the inhibitor, right? By activating the direct pathway, right? And then when I inhibit the inhibitor, right, I will have motor program starting, moving. And then I have, okay, motor program is on. When I want it off, I activate the indirect pathway, which is here, and the indirect pathway will end then in activating the red arrows or glutamatergic. So it will activate the inhibitor, and then I will shut down a motor program. Here it is the motor program commands on the brainstem, but in the textbooks you will find that this output is for the thalamus, and then you have neurons going from the thalamus projecting back to the motor cortex, and then you have your movement, okay? This is wrong. Well, this is not wrong really, we don't know, so some people are not agree with this loop, and rather they believe this loop is important. Both can be important. We really don't know, and it can be different in different species. But I just let you know. Okay, so these neurons here, to be activated, the ones from the direct pathway, they are very difficult to activate. You don't want to do, right? You don't want to activate a motor program just by chance, right? You don't want to do anything just because these neurons were activated randomly. So you have like a system to change the excitability of these neurons. These neurons are very, very hard to activate. So they need a lot of cortical input, a lot of thalamic input, and they need dopamine to put them in a state that they can actually respond to their input. And when dopamine is lacking, right, this pathway specifically doesn't work. So no matter how much input from cortex and thalamus, this doesn't work. And that is Parkinson's, right? So in Parkinson's, the dopaminergic neurons that provides dopamine to the stratum, they die progressively in many years. But when enough of them are dead, so we don't have dopamine, and we cannot start moving. And the L-dopas actually, L-dopa is a precursor, chemical precursor of dopamine. So if you give L-dopa to a person, they will cross the brain barrier, right? It will turn into dopamine, and the person will move back. And this is spectacular, right? It's a Nobel Prize discovery. But also, levodopa with time doesn't work too well. For many reasons, the L-dopa itself will just promote its these complications, but also because Parkinson's is progressive, right? And it's chronic, so it's just getting worse. It doesn't get any better in time. So people have been looking for other treatments, and one of these treatments is deep brain stimulation. And I think Manuel Guerrero talked about it yesterday a little bit. So in deep brain stimulation, you can put electrode, this time in the brain, right? In the subthalamic nucleus, actually. And you can then provide electric pulses to this. Here's how it looks in a real person, right? So one electrode in each hemisphere, each subthalamic nucleus, and you can provide this stimulation, and people get quite better. But then I will talk now why spinal cord stimulation would work, right, in this. And I will talk how the idea was generated, which it was actually on the wrong assumptions, right? But it turned out to be correct results, or the results were positive. So first, when people do the deep brain stimulation, they actually can record from the brain also during the surgery. And when they recorded in the Parkinson patients, they saw they have a very conspicuous, a very strong signal at 10 hertz. So this will be frequency, this is time, and these red like peaks are this actually, and that corresponds like to 14 hertz. So it means the local field potential, the electrical signal from this from subthalamic nucleus oscillates very strongly at 10, 20 hertz, more or less, this here. But when I start stimulating, I will actually shut down this conspicuous signal, and at this moment is when the person gets back into a better motor function. So, okay, so they say, okay, this is a biomarker of Parkinson's, so we eliminate this, right? It means that we can get back our motor function, right? By any means that you move this. And the second idea of this, the second idea for the genesis of this, is that this is epilepsy, this is a rat, this is the same signals, right? Electrical signals, local field potentials from the brain. This is the thalamus, this is a primary somatosensory cortex. And what you see here is actual epilepsy, right? And epilepsy looks like the same, right? It's like very strong oscillations. They're in lower frequency though, but it's very strong oscillations. And when you put an electrode in the trigeminal nerve of the rat, and you start stimulating, you will get rid of these signals, and you get rid of the epilepsy. So, and we just stop stimulating, the epilepsy comes back. So what does this mean? It means that you can change the oscillations, these pathological oscillations in the brain, not necessarily by putting electrode inside, but you can do it by stimulating peripheral, right, outside the brain. That's the message. So that was the idea that originated the experiments where we stimulated then spinal cord stimulation, also the trigeminal, on preclinical models, and later it was done in patients, and also I will talk a little bit about the potential mechanism for this. So this is a mouse with modeling Parkinson's disease, so it has very little dopamine on its brain, so it doesn't move. It cannot initiate motor programs, right? But once you start stimulating the spinal cord, when this turns red, now, there, so it will start, you know, after a little bit of accommodation, it will start actually moving around and do like mice, normally mice stuff. Okay, so which means this concept works, actually, right? If I stimulate the spinal cord, I'm freezing the inability of the brain to activate the direct pathway. But shortly after we published this, people started to try this in humans because the spinal cord stimulator, they exist for a long time. So there were there were stimulators, there were Parkinson patients, and they implanted the spinal cord stimulation, and it didn't work. So there were like 2 patients, I think, didn't work at all, failed, right? But at the same time, another group in France, actually, this doctor, he had a patient with Parkinson's, with chronic pain, and with a stimulator already implanted because he had chronic pain. And in this patient, it really works like very, very good. So let me see if it can play there. There, okay. Let me see. Here he's, oh, you cannot see the other one. okay, you will have to believe me, but here he is with the stimulator. So all his symptoms that should be in this one are gone, okay? Okay, but it doesn't matter. I have like 10 more videos. Okay, it didn't work. But there was the first patient. But what was the problem with this patient? That he had chronic pain. And where you're in pain, you don't move much, right? And when your pain is relieved, you move more. That is well known. So this patient actually, you could say, okay, but this is not like the spinal cord stimulation for Parkinson's. It's because he's in pain, and now he's not in pain. Okay. So then we started to do experiments in primates. So these are marmoset monkeys. And marmoset monkeys, they are very similar to humans. Of course, their brains are just very human. And they have like the same, of course, the same areas that we have for motor control. So what you see here, this is the brain, and the dot, which is not very clear in some of the pictures, indicates where in the brain we are recording. So you can record the primary motor cortex, you can record the putamen, which is the striatum, right, basal ganglia. GPI is also the globus pallidus internal, also basal ganglia, globus pallidus external, subthalamic nucleus, and the motor and the sensory thalamus. So you can put electrodes all at the same time, and you can actually measure what's happening in the brain. And what you see in a Parkinsonian marmoset is that it generates the 10 hertz peak the same as a Parkinson patient, just the same. And also you can see that when you apply the spinal cord stimulation, it's gone, right? But then when we saw like the average of this, it wasn't very good, really, like the decrease of the beta. These are called beta oscillations, right? It wasn't really like that much in everywhere, and it didn't. Importantly, it didn't match always like the physical recovery of the monkey. So instead of looking at the local field potential, we went to look at the rate of the neurons in these areas. So what you see here is that is the neurons, the firing rate of the neurons in each area. And in green is like no changes, red is like increase of the firing rate, and blue is decrease of the firing rate. And here you can sort according to the frequency of stimulation, and you see something very clear. That is, at low stimulations, especially at 14 hertz, you have a lot of excitation, right? So your neurons are firing more. But then at higher frequency of stimulation, you have less, you have inhibition, not just excitation, so it's just inhibition, no. So, and this is very good because, okay, I have like a correlate, electrophysiological correlate of the frequency I'm stimulating, but then it's bad because the results, the motor improvement was different didn't correlate with this. So I would expect that, for example, for this electrophysiology, I will have an improvement, and for this I wouldn't, but that wasn't the case. So that means neither local field potential, neither the firing rate can explain my results. So then we went for a third analysis, which is not looking at the beta oscillations locally, but to look at how the beta oscillations are correlated among the different areas, right? So it's called coherence. So for example, here I'm looking the coherence between, okay, this is thalamus and thalamus. This is sensory thalamus, motor thalamus, and when the stimulation is off, I have beta oscillations, and they are very coherent between the two structures. And when I have stimulation on, the coherence is gone. Although the oscillation still exists in each one, but the coherence between the oscillation is gone. So you can represent this data here like this. So each node here is a structure, right? Somatosensory cortex, motor cortex, putamen, thalamus, and subthalamic nucleus. And the lines joining these nodes, they are actual coherence. So blue line means low coherence in beta. Red lines mean strong coherence. So this is the intact group, right? Very low coherence between the areas of the brain. And this is the Parkinsonian group, so very high coherence. So I mean, when you're Parkinsonian, right, what happens is not that you have beta oscillations, but you have all the brain doing, synchronized in the same rhythm. And you cannot initiate your motor programs. if you give levodopa to the Parkinsonian group, you know, the coherence goes down, and this is the best treatment we know, right? Levodopa. The subject is moving again. And when you give spinal cord stimulation, what happened? Also, coherence is gone. And this correlates right with the motor improvement. Okay, so now we have a theory, a mechanism that could explain this. So in the meanwhile, a lot of people tried this in their patients, either patients with chronic pain that had already implanted stimulator, or either patients that had only Parkinson's and they were implanted with stimulator. So currently there are like all of the studies, right? They sum 86 patients, and 80 of them have improved the motor symptoms. So the important thing here is the selection, the correct selection of the patient. This is the most important thing. And this is all the studies, right? And I want you to look at the frequency of stimulation, and if you see the frequency is different in every study pretty much. Okay, this is mind-blowing because you would expect that a single frequency or a single range of frequency will work, but this is not the case, right? Like deep brain stimulation. Deep brain stimulation, the frequency that works best is 130 hertz. But this is not the case. So this is sort of, it's not only annoying, it prevents, right, from applying the therapy because we really don't know what is the best frequency or what is the frequency to start with. So, and I will show you here, and I hope the video works. These are patients with long-time Parkinson's disease. They have deep brain stimulators in their brains, but the deep brain stimulation doesn't work any longer for the axial symptoms. So the axial symptoms are the symptoms posture, affecting posture, and affecting gait. So you see this patient is very impaired, although the deep brain stimulation is on, right? She's very impaired on walking, if you remember the other video, right? Very short steps, and also freezing, right? But when we do the spinal cord stimulation together with the deep brain stimulation, now this person gets quite better on walking, right? So she can actually walk. There is freezing. This is freezing. She's trying to turn, right? This is a volitional movement, and it's totally frozen. So I will just look quickly. These are 4 patients from Brazil. So he isn't that bad, but he has freezing. Now you'll see the freezing. There, froze, right? And freezing is gone when you stimulate. So we'll go forward a little bit. Probably not. Okay, we'll just move to next. There are 2 more patients, but no need for. Okay, so. So what are the challenges and some of the take-home messages for this? This is a study in Canada, also with Parkinson, only Parkinson's patients. And here what they did, they use a mat that can sense the force of your steps. So what you see here is actually the step of the patient. And this is a patient. Here is without stimulation, on medication, so he has the levodopa, but you see the steps are very poor, right? Very poor gait. This is spinal cord stimulation with one setting, one stimulation setting, and this is spinal cord stimulation with a different setting. So now you see that this one works perfectly, and this one gets worse, right? So what is the difference between this one and this one? The stimulation frequency is the same, 60 hertz, but the pulse duration is different. This is 400 and this is 200. And only this tiny difference of 200 microseconds in every pulse makes huge difference on the clinical output. So this is a problem. Why? I'll tell you why. Because, this is a problem because we only, to simplify things, we have only 3 parameters here. So one is the pulse width, the one that was different here. This is the intensity, which is sort of easy to deal with. And this is the frequency, right? How many pulses I have in 1 second. And we have so many, just with these 3 parameters, there are so many possible combinations, right? Around 13,000. This is a calculation for deep brain stimulation, but it's similar. So there are so many possible combinations that we should try in every patient to just know which is the best one. Yeah, that is, it's impossible. Yeah, practically it's impossible to do this. So that is the problem. And currently, even for deep brain stimulation, which we have been doing also in 40 years, the determination of what is the right frequency, what is the right amplitude, and what's the right pulse duration is an empirical process. It takes time, and it can take a month, it can take 6 months, or it can take a year. Yeah? And even worse, once the person has already the parameters, it's good to change them once in a while, because the effect, the therapeutical effect, starts fading with time. And what happens is that physicians, for example, they can implant, I don't know, they can implant, like, let's say on the low point, like let's say like 40, 60 patients a year. It means every year he has 50 more patients to change the parameters, like, I don't know, every 6 months or something. So it's impossible, right, to manage this part, the part of, it's called programming the right parameters. So one of the solutions to do this, this is the adaptive stimulation, it's called. And here what you have a brain or a nervous system or the complete person, right? And you want to monitor, you want to sense, you put a sensor on them to acquire a variable, and with the variable you will do something, an algorithm, right? That will determine what, when, or what parameters can be used, right, and go back to the brain. So this will constantly be updated. It can be in milliseconds, in seconds, or in days, or in months. But you want this system, right, that senses what is going on and then correct the stimulation parameters to keep a variable in a certain range, right? You want a certain variable in a certain range. So it could be also motor function. So let's go then for a conclusion. And I apologize because they're in Spanish. They're supposed to be in English. But let's start. So first, spinal cord modulation, spinal cord stimulation can modulate the activity of supraspinal structures, okay? So we stimulate the spinal cord and we can see changes in our brain, and this has therapeutical value. At supraspinal level, these effects are all over the brain. They are not just in the primary somatosensory cortex. They're not just in the basal ganglia. They are all over the brain. We're hitting all the brain when we do a single pulse, and I didn't show the data, but you can just deliver single pulses of stimulation, and then you can do the evoked response, and you will see it everywhere in the brain. And finally, the underlying mechanism underlying to the therapeutical effects of the spinal cord stimulation for the chronic pain, which has been used for a long time, and for some of the motor diseases like Parkinson's. You can use it in other diseases too. They are pretty much unknown, right? Okay, so we don't know really how it works. And finally, and most important, these stimulation parameters, right? The combination, they are key for the effect, for the therapeutic effect of the spinal cord stimulation. And it's a pending challenge in the area. Not only spinal cord, it's the same for deep brain stimulation, right? So deep brain stimulation for dystonia, for example, takes a long time to program, and actually people die. Some of the dystonia are they're lethal, and people die just waiting for the right programming, because it's not easy to do. So that's a pending challenge. So I will finish now. I will just tell you that, of course, this is some of the results I showed. They were done in the States, in the lab of Miguel Nicolelis at Duke University. The monkey experiments were done at the University of Rio Grande do Norte in the International Institute of Neuroscience. And data analysis has been done mostly at Sweden, at Lund University, by the group of Dr. Per Pettersson. And currently, we are working here in Universidad de Chile, in Hospital San Borja also, with David Aguirre, who is a neurosurgeon. And yeah, he likes to put electrodes on people a lot. So don't go nearby him. And that's it. So thank you very much.
Thanks, Dr. Romulo, for your speak. And now we have time for a few questions. Do you have anyone? Okay, Thomas?
Yes, yes. It seems to be work better in the thoracic, low thoracic part. So actually, some people claim that what we are doing here is actually we're activating like the motor, like the motor part of the spinal cord, and generating or facilitating like the communication. But the answer is yes. I didn't talk about that because that's something that is a decision that you take before implanting actually. So it's sort of key. So for chronic pain, you put the electrode where you feel the pain. So if your pain is in the leg, then it's low. If your pain is in the arms, then it's higher. For Parkinson's, it seems to work better the lower you put it in the thorax. No, there's. I don't think there are reports of pain. There are. I mean, you can activate the pain fibers, but you need to go really strong with intensity to do that. Intensity is another parameter. No, it is known that once you are in a certain configuration, then the effect will start fading with time. So it's better. It's like for the same money of the implant and all the hassle. It's better to keep changing the frequency. The brain adapts to the frequency because. So thank you for the question because this is very important. So this is very dumb, right? Ta 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 all the time. So the brain is made to ignore that because there's no information there anymore, right? When it starts, it's the same thing, so the brain will tend, the circuits, all the brain would just tend to ignore that. So you need to change the parameters in order to keep like the therapeutical.
Very nice talk. About the currents. The currents, if I well understood, it's the fact that you're going to stimulate in phase two different things that the vector you're talking are distant. So they are not arriving in the same point. I mean, you have a distributive convolution or constribution. So is it possible to envisage that, for example, you do stimulations? I know that practically it's not possible, but I mean, imagine that you stimulate the striatum in the time at t0, you can delay another part, and then in this way you can make a merge of and use a artificial current
To
summarize, to arrive in the point, a delay that makes that you have a
Okay. Yeah, this is a very good question. Yeah, we have looking for that for for a long time. We didn't find it yet.
So
yes, yeah. The problem is with that is that this is not a static circuit, so it is being modulated. So it depends, and we know that it depends on the state of the subject. So it's not the same stimulating when you're moving and doing stimulation because then the times, the delays will be different. And we have been working on the lags to different structures, and they vary a little bit more than that we would like. But yes, we think this is key in order to get this to work.
And the method that you show that there is a broad band of frequency that you use, is it possible difference of frequency due to this emergence of coherence or disappearance of coherence?
Yes, yes. Actually, it seems to be that each patient has its own best frequency. And the same happens with monkeys. That's very interesting. With rat, it's not the case, but it happens in primates. So yes. Yes. Yes, that's the adaptive. Yeah, that's the adaptive approach. But the adaptive approach has a problem, and no one has tried this because they all work on actually on having like this interface for decide when to deliver the stimulation. But again, the parameters are already set. So just when so what they do, they save energy, which is important for the pulse because you have to change it or you have to recharge it. But not for establishing parameters, and that's something we definitely want to do with the algorithms. It's just raw power of computation. Yeah, it's we have used like AI, but they're quite limited actually. So we use them for classifying signals, and that works pretty well. But when you start putting more the problem with artificial intelligence is you need a lot of data to feed it, to train it. Yeah, you're right. But the limiting thing there is to get the information from patients. But because for training like an AI, you need like thousands of patients. Yeah. And now this is available probably with the Michael J. Fox organization. They have like databases for like thousands, maybe hundreds of patients. So probably something that we could do in the future, yes, in the near future. But with those databases. By itself, we cannot get as many patients needed for AI.
So the network is stimulating bilaterally.
Yes, not exactly. It's very tricky. The neurosurgeons they just change them and they ask the patient where does he feel the paresthesia. So you want something symmetric, but they're not always symmetric. But okay, here. So this one have like one side, middle line, and the other side.
So what happens? So the stimulation is bilateral.
It's bilateral. Yes, it's bilateral. Should be bilateral. Should be.
But what happens if it's not bilateral for the patient? I mean, just by curiosity, they have.
I don't know because they never do it unilateral. They will actually change this. They will play. This is another complication that I didn't talk. So you have many. You can choose what are positive and negative. So they will move those until the patient feels like the paresthesia in both legs. Yeah. Yes, it does. It does. So two things happen. So L-DOPA is good but you want to take low doses always, because it gets worse. The more the L-DOPA you take, it gets worse. So you want to reduce your consumption of L-DOPA. When you combine it, when you use deep brain stimulation and when you use spinal cord stimulation, the effect adds to the L-DOPA. So you can actually, when you have deep brain stimulation or spinal cord, you can reduce L-DOPA and you can prolong the benefits of L-DOPA over time. So it's a good combination, actually.